want to play with it? All right. Okay, we'll get started. Good morning, everyone. Welcome on the second day of the 18th Annual Needham Healthcare Conference. Delighted to introduce Kezar Life Sciences. This morning we have Mark Belsky, CFO, giving us an overview and update, and also Chris Kirk, President and CSO, for questions. I'll hand it over to you, Mark.
Good morning, and appreciate you attending our presentation this early in the morning. As mentioned before, I'm joined here with two of our colleagues, Chris Kirk, who's one of our founders, is co-founder and the CSO, and Celia Economides, who's our Senior VP for Strategy and External Affairs. As you can see, the lawyers make us put up a forward-looking statement disclaimer, and I'm sure all of you want to read this morning, so I'll keep it up here for about 5 minutes. So Kezar has a multi-pronged investment thesis. Kezar is best known for our lead program targeting the immunoproteasome. We believe we have a novel, differentiated approach. KZR-616 is a first-in-class selective immunoproteasome inhibitor, and it has broad immunomodulatory activity and avoids immunosuppression. We're excited about the platform potential for KZR-616. We believe it to be a pipeline in a drug, and while this phrase is well overused in our industry, we believe we have a well-defined and aggressive development plan for its use in both rare and large market autoimmune indications. We also have a separate and distinct protein secretion program targeting the Sec61 translocon, which has the potential to generate novel drugs, potentially in oncology. We expect this year to nominate a clinical candidate. Getting back to the 616 program, interesting proof of principle data has been generated in off-label use of Velcade in severe autoimmune disorders, including lupus nephritis. I have a slide later showing all of the off-label use of Velcade. Recall that Velcade is a non-selective dual proteasome inhibitor. However, chronic therapy is precluded by toxicities driven by dual proteasome inhibitors. KZR-616 has a distinct safety profile from these dual proteasome inhibitors. In a phase 1 trial, KZR-616 achieved its desired PK, PD, and safety objectives and demonstrated anti-cytokine effect. During this quarter, we'll report initial results from the first 2 cohorts of the phase 1b SLE trial, and will announce enrollment of patients exclusively in a phase 2 lupus nephritis trial. We're well funded for trials in up to 4 additional autoimmune indications beyond lupus nephritis. We're not just a lupus company. We have enough cash to do all this, and we ended 2018 with $107.4 million in cash equivalents, and marketable securities. We have an experienced management team, and we have a strong board with drug development and finance experience. As I mentioned, we're not just a lupus company. Our first phase 2 clinical trial is focused on lupus nephritis that, unlike SLE, has quantifiable endpoints. We'll report on the first 2 cohorts of the phase 1b trial later this quarter. Shortly thereafter, we'll initiate phase 2 trials in dermatomyositis and polymyositis, which have high disease burden, and then potentially 2 other orphan autoimmune indications with unmet need. In our protein secretion program, we're close to determining our lead oncology indication, and we'll announce this later this year. We had 2 posters on this program at April's AACR. Our programs target protein homeostasis. KZR-616 is a selective immunoproteasome inhibitor. This is a unique approach and builds on well over a decade of work across multiple companies led by our co-founder and CSO, Chris Kirk. We're attempting to achieve the holy grail of immunotherapy by blocking immunomodulatory activity without being immunosuppressive. We believe KZR-616 to be active in a broad array of autoimmune disease models. Our other platform focuses on the Sec61 translocon. The 5,000 to 7,000 secreted and transmembrane proteins must pass through the Sec61 translocation channel. We have two approaches to disrupt this, with applications in oncology, immuno-oncology, and autoimmunity. And it's potentially an orally bioavailable small drug replacement for biologics. I'll have more on this later. This is very novel biology, and we have a leadership position here and in immunotherapy. First, let me just give you a quick history lesson. KZR-616 origins can be traced all the way back to proteolytics, where Chris Kirk was hired on day one as its first scientist to work on carfilzomib. Proteolytics initiated programs for selective immunoproteasome inhibitors, leading to ONX-0914, which became the tool compound for KZR-616. The intention was to discover another cancer drug. Chris, who's a trained immunologist, and his team quickly determined that this was a powerful immunomodulator. Prolific publication of papers occurred during the next several years. Kyprolis was approved in multiple myeloma, and while at the same time, Velcade off-label was shown to be active in patients with SLE and lupus nephritis. Amgen then acquired Onyx for $10.4 billion. Chris left Amgen, acquired the IP for ONX-0914, and with John Fowler, formed Kezar Life Sciences. KZR-616 has the potential to address significant unmet needs in lupus nephritis and myositis with significant commercial potential. These are very serious life-threatening diseases. No new effective therapies have been approved in the last 50 years. What drove us to lupus nephritis is the distinct quantifiable endpoints, unlike SLE, which is a more difficult regulatory endpoint, and there were no approved drugs. This is a common thread with dermato and polymyositis. Both have high disease burden and all have the classic hallmarks of unmet needs and inflammation with prolonged steroid use and all the side effects. So let me take a moment to describe immunoproteasome biology. Proteasomes are found in all the cells of the body. Proteasomes regulate intracellular protein degradation. There are two main forms of the proteasome in our bodies. The main form is the constitutive, which is found in all cells. The much rarer form, and found only in the immune system and at sites of inflammation, is the immunoproteasome. Drugs you know well, such as Velcade and Kyprolis, hit both forms, and in particular the beta-5 and LMP7 sites with cytotoxic effect. KZR-616 is different. It targets the LMP7, LMP2, and MECO1 sites, and it avoids beta-5, and all the while avoiding its toxic effect. KZR-616 selective inhibition of the immunoproteasome is not cytotoxic, and it results in broad immunomodulatory activity. The dual targeting proteasome inhibitors, Velcade and Kyprolis, result in cell death induced by protein buildup. However, our scientists at Kezar discovered that selective inhibitors do not cause cell death. As you can see on the right-hand side, KZR-616 has broad immunomodulatory effect on both the adaptive and innate immune system. It blocks inflammatory cytokines with downregulation of TNF-alpha and IL-23. Tregs are upregulated and B cells are downregulated. This slide is our proof of principle. Despite Velcade inducing cell death, a few clinical investigators explored Velcade off-label in immunology. The most well-known case was in Germany. What was exciting to us in the off-label use of Velcade and in patients with severe SLE and lupus nephritis was the rapid and sustained drop in SLEDAI measurements and median proteinuria. There was a 100% response rate and prolonged remission after treatment. However, Velcade was not developed for chronic autoimmunity disease due to peripheral neuropathy, its off-target effect. As soon as Chris and his team of scientists heard about the off-label data, they wanted to see if they could replicate the Velcade data with immunoproteasome inhibition. In a nutshell, they did. They were able to match Velcade's efficacy using only 1/3 of ONX-0914's maximum tolerated dose. This slide details multiple published accounts of clinical activity with Velcade in autoimmune indications, both rare and large market, and where we have generated preclinical data with either ONX-0914 or KZR-616, showing we could potentially go with KZR-616. This is a reason that we believe we have a pipeline and a drug, and importantly, why we believe Kezar is much more than just a lupus company. A bit more on animal data we've generated over the years. In this 2009 mouse model of ONX-0914 and a mouse model of RA, in the upper right-hand side, there was separation from vehicle after the first dose. There was reduced expression of inflammatory cytokines. The subunit inhibition profile was good, and we saw the desired inhibition levels of LMP7, LMP2, and MECA1 while avoiding excessive inhibition of beta-5. You'll see later we were able to replicate this with KZR-616 in a phase 1A healthy volunteer study. In another mouse model, we compared ONX-0914 to the blockbuster drug Enbrel. ONX-0914 had a deeper anti-inflammatory response relative to Enbrel. Next, let me show you some preclinical data with KZR-616 compared to MMF or CellCept. In this mouse model, KZR-616 beat CellCept head-to-head and had prolonged effect. When combined with CellCept, there appears to be synergistic effect. The combination of KZR-616 and CellCept was better than just CellCept or KZR-616 alone. As a result, our phase 2 trial will test KZR-616 in combination with CellCept against CellCept alone in lupus nephritis patients. Finally, in multiple animal models and data from thousands of Kyprolis and Velcade patients demonstrated that immunoproteasome inhibition is not immunosuppressive. We did not see immunosuppression in the animal models. The animal models had normal antibody levels with KZR-616 therapy, and we did not see the toxicity seen with Velcade or Kyprolis. Now let me turn to some human data. We ran a robust, healthy volunteer trial with 82 patients. Our goal was to demonstrate selective inhibition of the immunoproteasome and characterize PK and safety data. We dosed both subcutaneously and IV in both single and multi-dose portions. We only treated for 1 month because treatment with Velcade or Kyprolis could only be tolerated for up to 1 month. Now let me talk about safety data. We saw the safety data we expected. There were no hematologic adverse events which manifest in Velcade and Kyprolis within 1 month. Additionally, there was no peripheral neuropathy, which is an off-target effect of Velcade. The most common AEs related to injection site reactions. Most were mild and transient. There were no severe ISRs or ISRs resulting in discontinuation. We did not notice at the highest level of 60 mg, but we did notice at the highest level of 60 mg that 4 of the 12 subjects experienced a systemic drug reaction. All were grade 1 and 2. The 2 considered SAEs were due to hospitalization for observation, and these were similar findings reported for Kyprolis. We're pleased with the KZR-616 results. It had well-behaved PK and was 100% bioavailable. From a PD perspective, we saw what we expected. We saw 80% inhibition at doses greater than or equal to 30 mg, greater inhibition of the immunoproteasome relative to label doses Kyprolis and Velcade, and inhibition of the constitutive proteasome did not exceed 40% at any dose level. We tested in doses of 30, 45, and 60 mg. In all doses, and in all of these 3 dose levels, KZR-616 hit the desired target profile. It avoided excessive inhibition of beta-5, and it achieved the desired inhibition levels of LMP7 and LMP2. This was similar to the data that I showed you earlier in the animal model. Because the phase 1A was a healthy volunteer study, we could not look for efficacy, so we looked for bioactivity. We took blood samples from the 45 mg dose cohort patients to measure ex vivo cytokine release. The drugs showed bioactivity in the ex vivo assays, and because of these data, we felt 45 mg was the correct starting dose for the bridging phase 1B trial. And this brings me to the current trial and what you can expect over the next coming months. In an open-label study, we started with a 45 mg dose measuring safety, PK, and PD. We then moved to a 60 mg cohort. Once we have safety review of the tolerability at 60 mg, we're going to start a randomized, 3-arm, placebo-controlled, double-blind trial to evaluate the safety and efficacy of KZR-616 in lupus nephritis patients. Patients will be randomized equally to one of 3 arms, testing 2 dose levels or placebo. All patients will remain on their CellCept and prednisone therapy during the 13-week treatment period. We'll have a full panel of quantifiable measures, including complete and partial renal response rate. Again, we'll also be reporting initial data from the 45 and 60 mg cohorts later this quarter. Our major focus over the next 7 quarters will be on the lupus nephritis trial. We may explore different dose timing and schedules to learn more about the drug in the phase 1B. We recently announced a phase 1 healthy volunteer study testing a new lyophile formulation, which we believe will be more patient-friendly. We're going to launch phase 2 trials in DM and PM, and possibly 2 additional autoimmune indications to further demonstrate our activity of KZR-616 in multiple indications. This is why we believe that we have a pipeline in a drug. In parallel to our 616 program, we're working on two distinct small molecule approaches to modulating protein secretion at the Sec61 translocon. All 5,000 to 7,000 secreted and transmembrane proteins must pass through the translocation channel. And we're taking 2 approaches to this. We're either going to block a broad assortment of proteins that can potentially drive cytotoxic tumor death, or targeted approach of 1 or a handful of proteins, much like a biological. And this is quite novel. So let me recap. KZR-616 is a novel differentiated approach. We have the potential to be a first-in-class agent with a promising safety profile and the potential to avoid immunosuppression. KZR-616 has the potential to be a pipeline in a drug. We're excited to prove this with data in multiple indications we'll be pursuing. Our protein secretion program, which you'll hear more about this year, offers significant potential to generate novel drugs. Velcade has been a big part of our KZR-616 story, and it's been a nice proof of principle. The phase 1 KZR-616 safety data has been what we expected. And then the initial data from the first 2 cohorts of the phase 1B SLE trial will be disclosed this quarter, along with the initiation of a phase 2 trial in lupus nephritis. Finally, we expect to initiate trials in up to 4 additional autoimmune diseases later this year, and we're well funded to do all of that. Thank you for your attention this morning.
Obviously have a couple of cohorts coming up, and you guys can talk a little bit more about how we disclose what we should look for next week.
So it's our goal to present data that we have at a major medical or research conference. That's going to be Kezar's MO going forward, and this first study is certainly no exception to that. So the hope is to be able to present these data at a research conference this quarter. We can't obviously say which one at this time until there's release of abstracts, but that's the goal, and we hope it to be, and plan it to be a rather fulsome data presentation.
How would you define success versus what should you look for?
I think what I would guide folks to look for is first safety of KZR-616 in a patient population. We really look at the 1B as an extension of our healthy volunteer study. The goal is to determine that weekly administration at what we believe to be therapeutically relevant doses are well tolerated in a patient population. Because it's a non-randomized study, it's going to be hard to look at the efficacy data and be able to compare to historic data sets, say for Benlysta. However, we are doing an extensive look, including SLEDAI and organ-specific scoring systems like the CLASI and the swollen and tender joint count. And if those are going directionally in showing signs of improvements, along with physician and patient global assessments, I think you can look and see that there's directionality in terms of the response to 616 in these patients, and that hopefully will bode well for the randomized phase 2 data in lupus nephritis, which is the real meat of the study.
You mentioned a few times that you want to be more than a lupus company, so you said that you want to look at maybe 4 other indications. Some of them I imagine you don't want to disclose at this point, but maybe you can give us a sense of direction where you were thinking, or what types of indications, what would inform your decision, that sort of thing.
Yeah, so we've been extensively looking at the autoimmune unmet need disease space, in particular orphan indications for which there's a patient size that's roughly equivalent to either the lupus nephritis patient population that we're going after, which is roughly 100,000 U.S., or the myositis population around 75,000. So patient size matters. Quantitative endpoints, whenever possible, matter for clinical trial design. A defined regulatory path for us. Competition makes a difference. We don't want to be in too crowded of a space this early on. And then finally, if there's Velcade precedent data, even if it's just a handful of patients that have responded to Velcade therapy with this disease, that's going to be a strong indicator for the potential for KZR-616.
Would you run all, I guess it would be 5 indications all in parallel, or are you planning to stagger these? Some of them would be label expansion, or would you look at all 5 of these indications in parallel?
Yeah, so the goal of these phase 2 studies is in a randomized fashion, show whether KZR-616 has activity in these autoimmune indications. If we see success, we'll look to do full development in these indications. The hope is to be able to ultimately make KZR-616 a drug with a label that contains multiple indications, which of course is the case for many biologics in the autoimmune space.
You also mentioned you're going to have a formulation with the story around that. I think it was a lyophile formulation, but behind that work.
Yeah, when we started clinical studies with KZR-616, the goal was to get into the clinic with the formulation as quickly as possible, and one that gave us enough flexibility for understanding the dose levels that we'd want to take forward. And so what we went with was a frozen liquid. We know that that's not amenable to patient self-administration for a subcutaneous drug like KZR-616. So we've developed a new formulation. We had already checked with the FDA that we could switch out for this new formulation in the phase 2 study in lupus nephritis, and we will do that and put this formulation in all new studies. But we felt it best to get some PK and PD data in healthy volunteers. So we went back to Australia, the very same site, in fact, that we ran our initial healthy volunteer study to gain this information, which we think, one, helps guide us in ultimately getting to patient self-administration, but also might be useful for European regulators who might want to see bioequivalency data before allowing us to start trials in Europe.
Another thing I wanted to ask you about, you had a couple abstracts you mentioned recently. You want to just give some highlights around data you presented there?
Yeah, we had 2 really well-received posters at AACR. One was focused on the potential of protein secretion inhibition and included molecules that could block multiple cytokines simultaneously, both in vitro and in vivo, molecules that could block multiple immune checkpoints in vitro, and then molecules for which we have derived a high degree of selectivity for an individual therapeutically validated target, as Mark mentioned, a potential replacement, small molecule replacement for a biologic. The other poster was around our lead series, one from which we anticipate nominating a clinical candidate by the end of this year. These molecules have very broad and potent anti-cancer potential, particularly in models of solid tumors like pancreatic, small cell lung cancer. These molecules are active in vivo, at well-tolerated and infrequent dosing, mediate rather potent anti-tumor responses. As far as we know, we're the only ones working on the Sec61 translocon, trying to inhibit it for therapeutic benefit, and we're really encouraged by the early data, and as Mark mentioned, we do plan to nominate a candidate by the end of this year, making Kezar a two-product company by the end of this year.
Okay, well, thank you all very much.
Thank you.